So before I start, I think I should thank Bharat for organizing this, and particularly Dr. Premnath and Sadhana Devanshi. These guys are really outstanding, as we go through for this particular event. Hopefully everyone can see my slide. Uh,
Yeah.
this is the outline of our talk. My name is Anurag Rathore. I'm a professor at IIT Delhi. Little bit about IIT Delhi. I think most of you in India know about it. IIT Delhi is one of the eminent engineering institutions that we have. It is considered the Institute of Eminence of the Government of India. But more importantly, IIT Delhi historically has been very active as far as collaborations with industry is concerned. So for many years, people at IIT, even though IIT Delhi is not considered, that we don't have that much industry here in Delhi, but the IIT faculty has been fairly active with respect to industry collaborations. And as it says here, more than 1,000 projects have been executed in the last 3 years. But my first guess is around 400 crores. So that quantum of funding has been coming to us. I work, besides being a professor in the Department of Engineering, I'm a coordinator for DBT Center of Excellence in Biopharmaceutical Technology. And I will not go into my own introduction. I think Sajani already covered that. But our lab, as you can see here, we kind of focus on processing and biosimilars, right? So when you think of biosimilars, questions that come to mind are obviously making a clone, making a process, and then of course characterization of the product to establish biosimilarity. And those are the activities that we are engaged in. We work with industry quite a bit, mainly because the mandate of my center is to create best practices for the biopharmaceutical industry. So you can see here that you see all kinds of collaborators. We have regulators, in terms of FDA, CDSCO, where we collaborate on research projects as well as on policy matters. We have vendors, companies like GE and Paul and Sartorius, and then we have, of course, the Indian biopharma industry as well as global biopharma industry. So you see all kinds of names there, and these are companies that we have worked with, in the last 5 to 7 years. We publish quite a bit because obviously being a research lab, our mandate is also to do cutting edge research, do cutting edge science, and we are fairly active that way. I have a very bright group of team members with some 20 PhD students and 20 postdocs. And this 50 people team, I would say we are organized like a biotech company. We are basically a small biopharma company where we are working on these activities. Just to give you, I don't expect you to be able to read everything here, but just to show you that there are a whole bunch of technologies that we have created. These are some that I can share, the names publicly. And then a lot of patents that we have filed, right? So patenting obviously is important because if you are doing something which is cutting edge, then to commercialize it, obviously you have to patent it. And so you can see here that fairly regularly we have been creating technologies and we have been patenting them with the idea to commercialize them potentially. This is I think some photographs that from our lab, and I should mention that this one here is basically our continuous facility. So we are, I think to my knowledge, we are one of the very few academic labs where we have a continuous facility set up, a lab scale facility. So we can do continuous processing for either microbial or mammalian products. Okay, focusing on the topic of today. It is what technology offerings we have. The first one is biosimilar for trastuzumab. Now, trastuzumab, those of you who are not familiar with it, it's a mAb, it's an anti-human epidermal growth factor receptor 2, and the originator is Roche, right? So Roche, and actually the more popular name that you might have heard of is Herceptin. That's Roche's product. So it has been around for a while. It's a blockbuster, multi-billion dollar sales every year. The patents have expired. You can see here that 2019, 2014, and the product is essentially for a specific kind of breast cancer, but for most breast cancer patients, this is the category that we are in. How is the market growing? So the market is growing fairly nicely, as you can see here, and I think in terms of obviously when you have this kind of product sales, you have multiple global players and you have multiple Indian players as well. So these are some of the major players that are making this product as of today. Now the question is, then why even create a biosimilar for it? Because the reason for that is that still, despite all these players and everyone, the cost of the product, both in the US or North America in general and India, is still quite high, right? How many Indians can actually afford, a therapeutic product at $15,000 per patient, right? Very few. And that's really their motivation to work on this, because at least we feel that, there is quite a bit of a change that can be, done in terms of affordability of these products. And in that context, I want to say that, we're not offering just biosimilar, but really, with that in the package is the ability to do continuous processing. And the motivation for that is that as per our calculations, it will result in almost a 70% reduction in cost of manufacturing for clinical batches and more than 35% for commercial batches. So it actually will impact your cost of manufacturing significantly. Very briefly, what is the process that we have created? And this is again our continuous process platform. We have our cell culture followed by acoustic-based separator, depth filtration, surge tank, continuous chromatography 1, continuous chromatography 2, continuous viral maturation, continuous chromatography 2, and then finally your IIT, right? So that's kind of our platform. Like I said, we have already demonstrated that this can result in a substantial reduction in your manufacturing cost. And I also want to say that this technology we are using here, this one, for continuous viral clearance, that's also our own patented technology. So this is one way to do viral clearance in a truly continuous manner. A little bit of text about the technology. So we are getting about 2.5 grams per liter in terms of titer, and the purification yield is about 23%. Which, if you talk to anybody from industry, they will tell you it's quite high. Now, when you're making a biosimilar, everything else is fine, but ultimately, the most important part is obviously biosimilarity, right? Because if it is not really similar to your innovator product, right, it's not going to go anywhere. So we have done quite a bit of biosimilarity assessment, and as you can see here, this is showing you the impact mark analysis, peptide mapping, fingerprinting, and you can see that our data is matching the Herclone, which is a raw product, quite well. Similarly, for physicochemical characterization, using SAR UV3D, using fluorescence, your reverse phase side fusion chromatography, and then finally your cation exchange chromatography for charge variant. So in all of these things, and this is showing you your charge variant profile, it is very similar to the innovative product. We've also tested functional separation because obviously, if if your product is not functional, it is useless, right? So this is really important as well. And as you can see here, that there is no statistical difference between our product versus innovative product as far as functional separation is concerned. This is just showing you a summary of everything that we have done with respect to biosimilarity. All these attributes that you see are, the typical attributes that any biotech company would look at. And that's one of the benefits, I would say, of our center, that we have the capability, all these tools that you see here, they're all in our center. The ability to do all of this, just by ourselves, that puts us in a very big advantage. So this is where we are. Like I said, the process development is over. We have the platform set up. We have demonstrated the process of preparing the test scale. We're getting a titer of around 2 to 2.5 grams per liter. Our purification yield is around 53%. And then, like I said, the cost of manufacturing is substantially lower. And I would like to say, in terms of the scale that the organizers created, we are basically here, right? That we have obviously not done any animal studies. That would be the next thing for us to do. Shifting gears to the second product that we have, ranibizumab. This is also popularly known as Lucentis. That's another product that is the innovative product that's out there, and it has actually the Lucentis is a product from Genentech, and the Novartis also has a very similar product that is there, and their licenses have also expired recently and expiring in Europe recently as well. Now this is something which is a treatment for what is called as age-related macular degeneration. Where it has been shown that this particular antibody segment, so it's not a complete antibody, it's a fragment, but it has been shown that it is very, very effective for patients with this indication. This is also a multi-billion-dollar product. Already, I think the sales, this is where we stand today, but you can see the projected sales are much higher. And unlike trastuzumab, this one is not overly crowded. You only have 2 global players, and in India, Intas is the only player with an approved product in this area. Now, this is again showing you that still, this is much cheaper than trastuzumab, obviously, but still, even for this, the price of these things, because you have to take multiple doses of the treatment, is actually, still high. And actually the availability of our Indian product is also somewhat limited. So there is that need to come up with, a cheaper version basically of this product, and that would really improve affordability of this product. So this is again, we have a continuous process that's for making Lucentis. We have in the upstream process, we have your bioreactor, and the way we can make it continuous is by basically having multiple bioreactors that are operating in a staggered manner. Then we do refolding, in using this continuous flow reactor. This is again our patented technology, and as well as precipitation. So we are doing this continuous refolding followed by a precipitation and a CFIR. And then again we have basically 2 chromatography steps in the process. So using this, what we are seeing is that we were able to one thing we were able to achieve is a very high refolding yield. Now, those of you who have worked with this product would know that it is actually very difficult to refold it. Most industry people, whoever has tried, is roughly around 15 to 30% range. And we have been able to, through optimization, we have been able to substantially increase that. And also again, just like the previous product, the fact that we are doing continuous processing, it in this case it results in a very high reduction in cost, as much as 80% of cost, right? You can basically make it at one-fifth the cost that it would cost, it would be otherwise. Again, biosimilarities, and we did this using Extensive, which is the Novartis innovative product. You can see that in the smart is very aligned. Peptide mapping, is again pretty good. In terms of charge variants, again, charge variant in this case is mostly the main product, and that's what we have as well. And again, functional similarity using SPR is again quite similar. You can see the KB values are fairly close. So this is where we stand on biosimilarity. You see there are some pending things. So these are things that are in progress right now, but we are fairly confident that they'll also come out as okay. Again, we have developed our upstream downstream process. We have demonstrated up to 5 liter by vector scale. Right now, we're getting a title of around 0.2 gram per liter, which again sounds very low, but again, for people who are making these fragments, whenever you look at technology for making fragments, this is actually pretty high. A high purification yield, mainly because of our really high refolding yield. That's why we have that. And then, like I said, the cost of manufacturing has been reduced as well. And essentially, both of our products are standing at the same place, because we are done with process development and we are done most of our analytical and functional biosimilarities. I think we have a few slides on what else is happening. We are obviously working with on a bunch of other biosimilars. Insulin is a collaboration between us and Professor Rahul Bhambure and NCL. GCSF and PET-GCSF we have in our lab. Asparaginase is also a collaboration between our lab and Professor Rahul Bhambure and NCL. And then we are making also SSA, which is human serum albumin in 65 and Simlin, which is a kind of insulin. Which is also in progress. So you see that most of them are at different stages of software development, but they'll all be, they'll all have a timeline of, I would say, 3 to 6 months. So they're fairly close. This is our infrastructure. Like I said, we are like a small biopharma company. So we have a bacterial suite, mammalian suite. Both cases, you'll see that we have multiple reactors all the way from, say, half a liter to 10 liter scale. We have a fairly large downstream processing lab, and then, of course, very large analytical and functional characterization lab. So all this under one roof, and like I mentioned before, we have one of the major activities that we have done is to create this end-to-end continuous lab, right? So we have a continuous platform where both for microbial and mammalian products that we can run. And I think our interest was that, again, the two products that are represented today, or any of the other products that are listed there, if anybody would be interested in licensing them or co-development of them, we can do it. We can also obviously do co-development of any other biosimilar, because again, the whole setup that we have created can be used for any other biosimilar that a company might have. I think that's all I have, and again, my communication details are here, so is here. So either of us, if you're interested, you can let us know, and I think I'll stop there. Thank you for your time.
Thank you, Professor Rathore. While we will invite people to type in their questions as I start the discussion, I wanted to request you to talk through a little bit of how continuous manufacturing in such processes really has an impact on cost. And what does it do? What are the process innovations while the molecule is still the same as a biosimilar? What are the process innovations that you're able to do to improve the cost effectiveness of the drug?
Okay. So basically, the platform that I talked about, we have, it's in very simple way. If you look at each of these unit operations, they are, most of them are the same unit operation that you have in tech as well, right? So in a very simple way, it's all about connecting them to each other, right? So that you are not stopping anywhere and you are not, you're not wasting your time basically. You're wasting your manufacturing time because of that. And that's the main benefit, that because you're not stopping, because your product is continuously going from one step to the next, your productivity increases enormously. And by our calculations and our experience, because we have run these platforms now for multiple products actually. We have done it for two different maps. We have done it for a couple of microbial products as well. We have seen productivity increase of anywhere from 10 to 15%, or sorry, 10 to 15 times, which means that in a given facility, I can make, almost 10 times more product. And that, now from a cost perspective, the benefit is coming from partly from this increase in productivity, partly from the fact that you are able to use your raw materials, particularly things like membranes, things like chromatography, resins, right? These are the things that are really expensive, and you're able to use them to their complete lifetime, basically. So that helps you in reducing the cost. Now, when I say that it's a matter of just joining them, I'm really oversimplifying it, because in reality, when you actually do join them, you will run into many, many issues. And it has taken us almost 5 years actually to figure out, how to join them, and more importantly, how to control them. Because when you are running a continuous process, there's no room for error. In batch processing, you can always stop and investigate and then decide what to do, right? Here, if you have to stop again and again, basically, you will be having a huge loss of productivity, right? So you can't afford to stop, and that makes it actually very difficult to run this such a process, for a long period of time.
Professor Rathore, there are some questions on the forward path and the timelines. I just wanted to understand, from the place we are right now, say for example, trastuzumab, what are the next steps and how long do you expect it will take in case it is, reasonably well funded and you're able to move fast? What timelines do you expect?
Right. So, I mean, that actually timelines, I would say, is really not much to do with us. They are the timelines that any biotech company has to follow, right? Because from our perspective, both of the products that we have shown are kind of ready to go. Yeah. The next step obviously would be animal studies, and for that we would need to take this process and make material for doing animal studies. Then you have to do the animal studies, get the data, get again approval from RCGM, and then of course your human clinical studies, right? So I think, if I were to guess, it actually really depends on, it depends a lot more on the commercial partner than us from here on, right? Because all we can offer is technology support and the product that we have. These timelines, it could be a minimum of 2 years, and it could be a maximum of 5 or 6 years, depending on basically how smoothly these things go and how experienced the company is in doing it, right? If you go to a company which has had many products approved, they know exactly what to do, where to do, how to do, right? And then things will go really fast. If it's a company that has no clue what to do, you are liable to make mistakes, and then things can take a long time. So I would say 2 to 5 years, I would say, to get this into the market.
Okay. There's a request for clarification. What is the expression host for trastuzumab?
It's so Chinese and Sugar.
Okay. And there's another question on what is the refolding yield for trastuzumab?
Oh, so just ignore that. That's a typo that we got there. There is no refolding in trastuzumab.
Yeah, so that's it. We noticed around 200 the item difference between originator trastuzumab and biosimilars and MS due to primary sequence or post-translation modification.
Yeah, I don't know that. That one I'll have to find out.
Okay. One other, in the Indian process, what are the kind of clinical studies that are needed beyond, to get your regulatory approval? Would you know that?
Yeah. So typically, you would need to do after animal studies, you would normally still need to do some phase one. Since it's a biosimilar, maybe you can get away with, your single study, let's say 100% study, which studies at your phase one, phase two, phase three altogether. So it could be a single study if it is very similar to the innovator product. And I think if you look at the most recent version of the guideline, then that's what they are recommending. It's anywhere from 100 to 200% data is what you would need.
Okay. Can you broadly tell us what is the process by which IIT Delhi engages with companies?
So we have over the years, we have a couple of things we have done. One is that we have done technology development for companies. So a company may have a specific technical problem, with one of their products, and we have fixed that for them and then transfer the technology to them. That's one kind of collaboration. We have actually partnered with companies where, they are a startup, they have a clone, they don't have the capability to do process development and analytical and functional acquisition and all that. So that's another sort of collaboration that we have done. We are right now talking to, in fact, we have transferred one of our continuous platforms to Biocon, for example. So that's another thing. This is giving process platforms to companies that can then adopt it for their own product manufacturing. So those kind of collaborations we've also done.
Okay, great. So I think with that, I will stop with the questions, and I'll request Prajna to take over again. Thank you so much, Professor Rathore. Okay. Would you know about, before we continue, there's one last question. Would you know about also the regulatory process for other submissions in other countries? US submissions or animal studies required for a biosimilar?
Yeah. So if you are going abroad, then yes, India is actually one of the most lenient, regulatory system that we have. So if you go abroad, yes, you will need to do in abroad. They don't require large animal studies. That's actually one of the few major differences between Indian regulatory and international agencies. So you don't need large animal studies, but you need small animal studies. But then when you go there, you will still need to do phase one separately, and then most likely a phase two, phase three combined, but a much larger trial. The biggest difference is in terms of the size of the trial. That the phase two, phase three trial that you have to do, to go abroad, is much larger than what the Indian regulator requires. Which is obviously a lot expensive as well.
Yeah. Thank you very much. Thanks. Prajna, over to you. Thank you.
I think Dr. Chirmunu has, Yeah.
No, first of all, Anurag, a very, very nice, good presentation. One question I had which may be relevant to the listeners over here. One is about continuous manufacturing, the second is about scale, right? So we always, obviously, we can learn from how small molecules have done continuous business as a very success of continuous manufacturing and has been quite successful in that. So something, you don't have to answer the question, but something we could learn from the small molecules and how they've done continuous manufacturing so well. But the question specifically for you as it relates to the different customers or the different people who may be on the call, is when you use continuous manufacturing, your footprint of your manufacturing facility can be very small, And you can have, maybe hundreds of companies doing small manufacturing versus, a large company making 20,000 liter tanks. So if you can comment on that, that would be useful.
So yeah, so Naveen said that, I mean, because you're getting 10 to 15 times increase in productivity, you can play it two ways. One is that I have an existing facility and I can make, 10 times more products. That's one way to play. Second is that, okay, I'm setting up a new facility. And so now, I can do it at roughly, 1/10 of the footprint. So my size of my tank, size of my equipment, everything is shrunk by 1/10. So he's right that way. I do want to say that, it's a bit oversimplistic because it is actually quite difficult to run this. So that, that's the reason I have never, I don't see having hundreds of companies doing continuous processing because it's just, the level of rigor and the level of understanding and complexity is quite high, which is already high for biopharma manufacturing. So this is even higher than that, right? So that will limit, you can't have small companies doing this successfully. But technically, he's right. I mean, the way I think I look at it is that you will have a few, say 20, 30 companies, hopefully, with this expertise, and then they can have small plants within their mega plant, right? So you can have literally in one room, you could be making one product, and you can have many such rooms like that. So it changes the way you think of manufacturing. You won't be needing a 20,000 liter reactor, to make a MAB anymore. A 1,000 liter reactor, a couple of 1,000 to 2,000 liter reactors may give you the a huge amount of